Hi guys, so this is part 2 of bioenergetics. We finished part 1 last time. We're going to start with a quick review of bioenergetics before moving on. So let's just begin. Give me 1 second, let me just fix the settings. Okay. So we discussed what are photosystems. We discussed the different types of photosystem. We discussed cyclic and non-cyclic photophosphorylation. We discussed how ATPs are made, and so on and so forth. The one thing that I don't think I told you guys last time, and that is slightly important, is the sequence from how an electron goes from photosystem 2 to the final acceptor, which is to form NADPH. So I would request all you guys to please just put this down in your notes. This is important. Okay. So I'm just going to use this picture as a reference. Okay. So the electron first goes to photosystem 2, chlorophyll A. Okay. From there, it is passed on to pheophytin. The first electron acceptor is pheophytin. Okay. Then I'm going to also type it. Okay. So it's better for you guys to understand. So chlorophyll A, which is present in the photosystem 2, to get from chlorophyll A, it goes to pheophytin. To get, then it goes to plastic quinine. Sorry. Pheophytin to plastic quinine to cytochrome complex. And I'm going to draw a double line over here because over here is when the H plus ion actually enters the chloroplast. Okay. Complex. Moving on to plastocyanin. Furthering on, moving to chlorophyll. Photosystem 1, chlorophyll B. Okay. Further moving on to ferredoxin. And lastly, NADPH. Can you get? Okay, understood. So it is from chlorophyll A to pheophytin, from pheophytin to plastic quinine, from plastic quinine to cytochrome complex. So if we can over here, see this is your photosystem 2, chlorophyll A, that is your pheophytin, pheophytin to plastic quinine to cytochrome. Okay. Over here is where the H plus ion comes inside, cytochrome complex to plastocyanin to photosystem 1, photosystem 1 to ferredoxin and ferredoxin to NADPH. ठीक है, this is the chain of electrons. ये याद रखना, हो सकता है question है, हो सकता है ना है, but just make sure you know it. Okay? ठीक है. Let's continue. So we did over till here. We discussed the Calvin cycle. We're done with that. Take care. Now we're gonna do something important. Okay. Something called photorespiration. So what is photorespiration? Can anyone tell me? What is photorespiration? Anyone knows? Anyone? What is photorespiration? Any idea, guys? Come on, come on, come on. I see a lot of people, but there's deadly silence. Okay, so I got an answer. Thank you, people. So Abdul Sapur says using oxygen in presence of light when O2 and CO2 compete for binding. Okay, you both are partially correct. So basically what happens, when there is a hot or dry day, and stomatas like close the leaves that close the stomatas in response to conservation of water, this also decreases photosynthetic yield. Why? Because it limits the access of carbon dioxide, and thus oxygen increases and carbon dioxide decreases inside the leaves. Okay, let me repeat. When it is dry, when it is hot, when when more water is required by the plant, stomata close. Stomatas on the leaf close. When stomatas on the leaf close, what happens is access of carbon dioxide decreases. When carbon dioxide decreases, there is no ventilation. Oxygen concentration increases inside the leaves. Okay, this condition where oxygen concentration has increased inside the leaf favors a wasteful process called photorespiration. Okay. Uh, in this, what happens is it is an alternate in plants. To prevent this, you have alternate methods where plants have mechanisms which help in preventing this and not waste any resources. So if you look over here, when plants do undergo photorespiration, oxygen combines with the rubisco instead of carbon dioxide, forms a molecule known as phosphoglycolate. When phosphoglycolate is formed, what happens? It to be converted back into 3-phosphoglyceric acid. It has to give out carbon dioxide, and that is when 3 carbon dioxide are given out. These are already fixed carbon dioxide inside the RUB, which are given out. It's a wasteful process. The plant needs to go back. The plant needs to can fix this carbon dioxide, and the whole cycle will repeat. Okay? So to prevent this loss of 3 carbon dioxide, plants have mechanisms to prevent photorespiration. The first mechanism is present in C4 plants. Okay, in C4 plants, the light dependent and the light independent reactions are separated physically. Okay. The light dependent reactions and the Calvin cycle are physically separated, with the light dependent reactions occurring in the mesophyll cells, which are the spongy tissue in the middle of the leaf, and the Calvin cycle occurring special cells around the leaf veins. These cells are also called the bundle sheath cells. So what happens? Mesophyll cells where you have your light dependent reactions, and in your near the vein of the leaf, if you have your light independent reactions in bundle sheath cells. What happens is atmospheric carbon dioxide is fixed in the mesophyll cells to form a simple 4-carbon organic acid called oxaloacetate. If you can look over here, carbon dioxide from outside is used up by PEP, by an enzyme called phosphoenol pyruvate carboxylase, to form a molecule called oxaloacetate. The oxaloacetate molecule is then converted into a similar molecule called malate, which is then transported inside the bundle sheath cells, where the carbon dioxide is released and the Calvin cycle takes place. Okay, let's continue. So this whole process, yes, it's efficient. You'll have carbon dioxide, which is now present in bundle sheath cells for Calvin cycle to take place regularly. This is an energy expensive process. What happens? You need to have ATP. ATP is required to convert the pyruvate back into PEP. So thus it does take up energy. Okay, but the best part is it all gives us a constant supply of carbon dioxide in the bundle sheath cells, and that's why photosynthesis is continuously happening. Okay, photorespiration is prohibited. It does not take place because carbon dioxide is present in enough quantities for the Calvin cycle to go forward. Okay, this mechanism is present in about 3% of all vascular plants. Crabgrass, sugarcane, and corn plants are common in habitats that are hot but less abundant in areas that are cooler. In hot conditions, the benefits of reduced photorespiration likely exceed the cost of ATP, moving the mesophyll cell to the bundle sheath cell. So what we're trying to say over here is, even though ATP is used, it is really beneficial for the plant because of the fact that there is no carbon dioxide which is wasted, and uh, processes, bioenergetic processes are taking place as normal. Okay, understood? Understood, guys? C4 method, C4 plants. Great. Let's go. Then you also have some plants have a different mechanism called the CAM pathway, or the crassulacean acid metabolism. I'm sorry, I don't know how to pronounce that. Okay. These are in dry environments. Cactus and pineapples have this pathway. Okay. Instead of separating them as far as two separate areas of the leaves are concerned, in such plants, it is separated in processes of time. So some processes take place in the morning or daytime, some processes during nighttime. What happens is at nighttime, the camp plants open their leaves, open their stomata, allowing the CO2 to diffuse into the leaves. When CO2 diffuses into the leaves, it is combined again with PEP using the enzyme PEP carboxylase to form oxaloacetate malate or other organic acids. Okay. Now during the daytime, okay, the stomatas are closed. Okay, the compounds do not open their stomata, but they can still photosynthesize. So when sunlight is present and light dependent reactions are taking place, these organic acids, malate, the other organic acids, they release their carbon dioxide. And when they release their carbon dioxide, they make sure that they maintain a carbon dioxide concentration where photorespiration is prohibited and the Calvin cycle takes place as normal. Okay, the CAM pathway does require ATP at multiple steps. However, plant species that use CAM photosynthesis not only avoid photorespiration but also are very water efficient. The stomata only open at night when humidity is higher and temperatures are cooler. This reduces water loss from leaves, and the CAM plants are usually very dominant in very hot and dry areas like deserts. Understood. So one, basically your C4 pathway is differentiated in terms of space. The mesophyll cells are present somewhere else and the bundle sheath cells are present somewhere else. Whereas your CAM pathway is separated in time. Your stomata open up at night. So malate and all your other organic acids are made at nighttime, which then release carbon dioxide, which is present for absorption, or which is present for the Calvin cycle to go forward during your daytime. Understood, guys? Understood the difference between C4 and CAM pathways? Are we good? Great. So this is a simple table, okay, giving you differences between the C4 and the CAM pathway and C3. So separation of initial CO2 fixation and Calvin cycle is not present in C3 plants, which are normal plants. It is present in C4 plants between the mesophyll and the bundle sheath cells. And in CAM plants, it's between night and day. Stomata in your C3 and C4 plants is open during the day, whereas in CAM plants, it's open during the night. And C3 and C3 plants is ideally adapted into environments that are cool and wet, where C4 is present in hot and sunny environments, and CAM plants are pleasant in very hot and dry environments. Over here is another few examples of such plants: tropical grasses, corn, sugarcane are C4 plants, succulents, pineapple, agave. Agave, so you get agave nectar, which is a sweet nectar. Okay, it is CAM plants. They do have very high water conservation. All your CAM plants, they lose the least amount of water because the stomata do not open up during daytime. So this is all done with plants. Now we're going to go ahead and talk a little bit more about glucose. We're going to talk about glycolysis. We're going to talk about how does glucose break down, what happens, aerobic reactions, anaerobic reactions. Simple, easy stuff, not very difficult. Okay. So you have aerobic reactions with the breakdown of sugar in the presence of oxygen and release carbon dioxide and water with sufficient amount of energy. This type of respiration is known as aerobic respiration, and the organisms that perform this type of respiration are called aerobes. Similarly, the breakdown of sugar in the absence of oxygen is known as anaerobic respiration, and this type of respiration is performed by anaerobes. Human beings prefer aerobic area respiration, but can also perform to a limited extent anaerobic respiration. It is not very efficient in human beings, but there is a possibility to undergo it. Yeast, bacteria, some bacteria, gut parasites, annelids, roots of plants, and are all anaerobic. Those which never need O2 are at all are obligate anaerobes, and we've done this before. Obligatory parasites के थे facultative, remember? Obligatory anaerobes हमने जब वहाँ किया था, हमने kingdoms कह रहे थे, prokaryote, monoera, हमने किया था obligate anaerobes होते हैं, which definitely do not need O2, whereas facultative are which respire anaerobically, but can also respire without with the presence of O2. Benefits of anaerobic fermentation is the alternative term for anaerobic respiration. Okay, you have a lot of benefits of it, especially economic. It is a source of ethyl alcohol in wines, beers. Yeast is yeast anaerobically respires to form bread. Milk is converted to curd, cheese, lactic acid is produced. Flavors of pickles is due to lactic acid and acetic acid. Acetone is also produced as a byproduct of fermentation. So this is glycolysis. I'm going to skip it for 1 second. I'm just, because we're on anaerobic respiration, I'm just going to go over the two pathways of anaerobic respiration, which are important for us. In humans, you have the lactate pathway, and in yeast, you have the ethanol pathway. In your lactate pathway, you have the glucose, which it uses up to ATP, produces to NADH to form pyruvate, okay? And then moves ahead and forms lactate, okay? The net result is the production of 2 ATP only. Okay, similarly, in the ethanol pathway, ethanol is being produced and carbon dioxide is given out. Okay, humans, when there is a decrease in oxygen, humans undergo the lactate pathway. It is again not very efficient as far as energy production is concerned, but it is an alternate form of respiration present in human beings. Let's come to the more economically important for method of energy production. Lots of ATP is produced in this. It is known as glycolysis. It's known as aerobic respiration. The first step in aerobic respiration is glycolysis. Where does glycolysis take place in a cell? Guys, come on, tell me where does glycolysis take place? Very good, cytoplasm. Glycolysis does take place in the cytoplasm, the cytosol. if you have multiple steps, you have almost 10 steps of glycolysis to take place. You have a glucose converted to glucose 6-phosphate, moving on to fructose 6-phosphate, fructose 1,6 bisphosphate. Then it breaks up into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then 1,3 bisphosphoglycerate all the way up to pyruvate. What you guys need, okay, the first question that I'm always asked is, सर पूरा याद करना है या नहीं याद करना? तो बेटा पूरा नहीं याद करना, आपको जो याद करना है वो आपको essentially 4 से 5 चीजें याद रखनी है, ठीक है? I would recommend you guys have a lot of time, तो ये चीजें definitely याद करो. First याद करो the two reactions where ATP are used, ठीक है? The two reactions where ATP are used are the first one where glucose is converted to glucose 6-phosphate, and when fructose 6-phosphate is converted to fructose 1,6 bisphosphate. Hexokinase and phosphofructokinase is the enzyme. और एक चीज मैं बता दूं ग्लाइकोलाइसिस के बारे में, इस साल नहीं याद करना, ठीक है, whatever. Next year 100% याद करना पड़ेगा, because first chapter you're going to do in med school is cell biology and genetics, and this is like going to be your first topic. So तब तो याद करना ही करना, so might as well अभी याद कर लो. Okay, so these are the energy expending steps where energy is used up. ATPs are used up to form into the compounds. Glucose is converted to glucose 6-phosphate by the use of 1 ATP, and fructose 6-phosphate is used to form fructose 1,6 bisphosphate by using another ATP. Now this is a biphosphate 6-carbon compound, which then is broken up into two. There's aldehyde 3-phosphate and dihydroxyacetone phosphate. They're similar compounds of isomerization, so everything is converted to glyceraldehyde 3-phosphate eventually. Now, the first step is your phosphorylation, where an NADH is produced, then an ATP is produced, then you have another step where an ATP is also produced. So these three steps you have to remember. The first one is glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate, and next is 1,3 bisphosphoglycerate to 3-phosphoglycerate. NADH is produced and ATP is produced. And lastly, phosphoenol pyruvate to pyruvate, where another ATP is produced. Okay, so now at the end result of glycolysis, you have a glucose molecule, which is finally converted into pyruvate. Okay. Apart from that, two ATPs are used. Glucose is converted to two molecules of glyceraldehyde 3-phosphate. Each molecule of glyceraldehyde 3-phosphate will give you one NADH and two ATPs. One NADH and two ATPs. Okay, so when you have one molecule of glucose, you have two molecules of glyceraldehyde 3-phosphate, you will have two NADH and four ATPs. You have two NADH and four ATPs. Okay, each molecule of glucose to be converted to pyruvate will use up two ATPs, but in turn give us two NADH and four ATPs. The net gain of ATP in glycolysis is 2, because 2 are used and 4 are produced. 4 minus 2 is 2. Okay. If I have to write the reaction for this, what I will simply do is write glucose, okay, to which we have to add 2 ATP, okay, because 2 ATPs were added, okay. Plus we also need NAD as raw material, okay, and we need 2 NAD, okay, will give us 2 pyruvate, okay, along with 2 NADH, along with 4 ATP, along with 2 molecules of water. Okay, so one molecule of glucose takes up 2 NAT. ATP requires 2 NAD to be converted to 2 molecules of pyruvate, 2 NADH, 4 ATPs are given out in this process, and 2 molecules of water. Net gain is 2 ATPs. Understood, guys? Great. So now once pyruvate is made, once pyruvate is ready, it needs to be taken from a cytosol to the mitochondria for further processing. Okay, you have your transport proteins over here which take this pyruvate molecule to the mitochondria. And during the process, it also needs to be converted to another compound which will then be further used in your Krebs cycle. The compound is known as acetyl-CoA. Your 3-carbon molecule, which is your pyruvate, gives out a carbon dioxide, forming a 2-carbon molecule. Coenzyme A, which is another cofactor, is added to it, and NADH is produced in this process, and you have your acetyl-CoA. This process is known as pyruvate oxidation. Okay, this is simple. You have your pyruvate. Your pyruvate is taken inside the mitochondria to the matrix. Meanwhile, it is transported by a transport protein. It is decarboxylated, a carbon dioxide is given off, coenzyme A is added, and NADH is produced, and here you have your acetyl-CoA molecule. This acetyl-CoA is then taken for further processing to something known as your Krebs cycle, okay, or your citric acid cycle. Okay, this is extremely complex. If you can see lots and lots of circles, this is an easier one. Okay, this is a more difficult one, but I'm going to start with the more difficult one first so you understand, and then I'm going to go over the easy one. Okay, again, the same question, do we need to learn this? Again, I would recommend just learn the steps jahan NADH या FADH ATP produce हो रहा होता है, ठीक है? So you have your acetyl-CoA. Acetyl-CoA is first combined with oxaloacetate by the enzyme citrate synthase to form citrate. Okay. Citrate is then taken to form cis. Again, this is just a simple isomerization. Isocitrate banta hai. Isocitrate is the first time alpha ketoglutarate banta hai, or this is the first time where a molecule of NADH is produced using the enzyme isocitrate dehydrogenase. ठीक है? Also a carbon dioxide is given out. So जो आपका molecule था 4 carbon, ठीक है? If you can look here, sorry, 5 carbon. Sorry. 6 कार्बन मॉलिक्यूल जो था अब वो 5 कार्बन मॉलिक्यूल हो जाएगा, ठीक है? पहले 6 कार्बन मॉलिक्यूल था, 2 वहां से आए थे, 4 ऑक्सैलोएसिटेट से थे, 2 एसिटाइल-CoA से आए थे, टोटल 6 कार्बन मॉलिक्यूल बना था। अब वो एक कार्बन डाइऑक्साइड निकल गया और वो बना है 5 कार्बन मॉलिक्यूल, ठीक है? ये देखो 1 2 3 4 5 6 थे पहले आइसोसिट्रेट में, अल्फा कीटोग्लूटरेट में 5 होंगे, 1 2 3 4 5, ठीक है? Isocitrate dehydrogenase and NADH molecule is produced, a CO2 is given off. Similarly, alpha ketoglutarate dehydrogenase will act on alpha ketoglutarate to form succinyl-CoA. Again, an NADH molecule will be produced. Carbon dioxide will be given off. जो आपका 5 carbon था, अब वो 4 carbon हो गया, ठीक है? NADH molecule is made and carbon dioxide is given off. Then succinyl-CoA is made into succinate using succinyl-CoA synthase, and a GTP molecule is made in this process. GTP will then further form an ATP. Succinate will then be converted into fumarate, and by succinate dehydrogenase, and FADH2 molecule is again formed in this process. And fumarate would eventually then be converted into malate, and malate से फिर oxaloacetate बनेगा और इसमें malate dehydrogenase will act, forming another NADH molecule. So one turn of this cycle, guys, one turn of this cycle will give us 1 2 3 NADH, 1 FADH, 1 GTP, and 2 carbon dioxide. Okay, let's remember every molecule of glucose forms 2 pyruvate, which will then form 2-acetyl-CoA. So per molecule of glucose, this cycle will go on twice. So each turn of this cycle per molecule of glucose, that means the cycle will turn twice, giving us 6 NADH, 2 FADH, 2 ATP, and 4 carbon dioxide. Okay, a simpler view. Acetyl-CoAs form citrate. Citrate then goes multiple steps. This is very simple. यहाँ सिर्फ मैंने आपको क्यों दिखाया ताकि आपको number of carbons दिखें and you will see कहाँ क्या निकल रहा है. ठीक है? You have your NADH 1, 2, 1, 2, 3, एक FADH and 1 GTP or which will then be converted into ATP. Okay, one carbon is lost over here, the other carbon is lost over here. One carbon is lost over here, the other carbon is lost over here. Isocitrate to alpha ketoglutarate is where the first carbon is lost, and alpha glutarate to succinyl-CoA is where the second carbon is lost, both as CO2. Okay. So in a single turn, I'm just repeating myself, 2 carbons enter from acetyl and 2 molecules of carbon dioxide are released, 3 molecules of NADH and 1 molecule of FADH are generated, and 1 molecule of GTP is produced. These figures are for 1 turn of the cycle corresponding to 1 molecule of acetyl-CoA. Each glucose produces 2 acetyl-CoA, so we need to multiply these numbers by 2 if we want the per glucose yield. Okay. So now this is where glucose is completely finished. Our carbon dioxide ban gaya, finish ho gaya. Now all this NADH and FADH will then now be taken to form ATP. Okay. To produce ATP, NADH is very good at donating electrons in redox reaction, so it can transfer its electrons to the first complex. NADH is very good, so it transfers its electrons to the first complex. As electrons move through a complex series, it causes hydrogen ions to, wait. So if you can see over here, the hydrogen, this is the mitochondrial matrix, and this is your intermembrane space. Each, as electrons are moving on, it causes hydrogen ions to go from your mitochondrial matrix to intermembranal space. Okay? Yeah, sorry. As electrons move through complex 1 in a series of redox reactions, energy is released, and complex uses this energy to pump protons from the matrix to the intermembranal space. FADH is not as good in donating electrons, so it cannot transfer electrons to complex 1, but instead it transfers it to complex 2, okay, and so on and so forth. Beyond the first 2 complexes, NADH and FADH both travel the exact same route. Both complex 1 and complex 2 pass the electron to a small mobile electron carrier called ubiquinine. From ubiquinine, it goes to complex 3. As electrons move to complex 3, more ions are pumped across the membrane, and electrons ultimately delivered to another mobile carrier called cytochrome C. Cytochrome C carries the electrons to complex 4. When a final batch of hydrogen ions is pumped across the membrane, complex 4 passes electrons to O2, which split into 2 oxygen atoms and accepts to form water. If you can look over here, water is formed. Okay. Now, so once all of this is formed, once all of this hydrogen has been taken, is present in the intermembranous space, I think I've missed out a slide. Give me 1 second, guys. 1 second. Giving me 1 second. I've missed out a slide. I'm just going to show you guys a picture. Okay. Give me 1 second. Second. Okay, guys, can you see? So now all your etc. Okay, great. So the electron transport chain, what we did was the electrons were deposited by NADH and FADH into complex 1 and complex 2 respectively, causing H plus ions to go from the matrix to the intermembrane space. There are more H plus ions in the intermembranous space than the matrix. What will eventually happen once they're present in the intermembranous space, they will use a channel called ATP synthase, which is also an enzyme, to go back into the matrix. Once H plus ions are going back into the matrix, that is where ATP production happens. Okay, it's like a cycle. H plus ions, they charge and they give out a current, and that is where the energy comes from, and ATP is produced. So this is how ATP is produced using the ETC. Your NADH and FAD are give off their electrons to complex 1 and complex 2 respectively. From complex 1, it goes into different electron carriers, similarly with complex 2, causing hydrogen ions to go from the matrix to the intermembranous space. And then ATP synthase was the enzyme which will work to cause the hydrogen ions to go from intermembranous space back into the matrix, causing ATP production. Okay. Now, the last thing that we're going to do with this is the ATP yield per glucose molecule. Guys, don't get confused. There are two different things to this. I'm just going to go first through the interway. Okay? The interway, interbooks में ये दिया हुआ है, ठीक है? Glycolysis से you have a 2 ATP net gain, ठीक है? जो हमने कहा था 4 ATPs were produced, 2 were used, and you have 2 NADH production. Now, when NADH are produced, if you can see, glycolysis took place in the cytosol and mitochondria is inside. There are a few shuttles through which NADH needs to come in. And when those shuttles are used, 2 ATPs are wasted. So the net gain, instead of 3 ATPs per NADH, is 2 ATPs per NADH. NADH usually gives us 3, but because of the location, it will give us 2 now. FADH will give us 2 regardless. Then you have pyruvate oxidation, which will again form 2 NADH, resulting in 6 ATPs. And then you have a citric acid cycle. Per cycle, you have 1 ATP, 3 NADH, and 1 FADH. Per molecule of glucose, you have 2 ATP, 6 NADH, and 2 FADH, resulting in 2, 18, and 4 ATP production, respectively. We're coming down to a total of 36. Okay, this is what Interbook says. The A-level books are slightly different, and actually it's slightly different. Actually, so when your glycolysis produces 2 ATPs, 2 NADH will produce anywhere from 3 to 5. Why do they say is because every NADH molecule does not realistically give you 3 ATP. It gives you 2.5 ATP. And every FADH molecule does not give you 2 ATPs, but it gives you 1.5 ATP. Okay? So over here, because ATPs are also used, remember I just explained that NADH को अंदर आना होता है matrix में, तो इसीलिए there are different transfer proteins, कुछ में ज़्यादा ATP production use होती है, कुछ में कम होते हैं, so ये 3 to 5 दिया हुआ है यहाँ. Pyruvate में 2 NADH produced with 5 ATP. Citric acid cycle में 2 ATP, citric acid में 6 NADH, which results in 15 ATP अगर आप लगाओ 2.5 से, ठीक है? And 2 FADH, which are 1.5 ATP, like out to 3. So ultimate ATP yield actually is 30 to 32 ATPs. and 30-32 depending on the transport protein used for after glycolysis transport NADH from the cytosol to your matrix of the mitochondria. Okay. Ahan usually ऐसे questions नहीं पूछता, but अगर पूछे तो please I would recommend आप लोग 36 लिख दीजिएगा, ठीक है, because inter books में 36 है, कि according to 36 है, ठीक है. अगर तो वो ideal yield की बात करें then definitely 36 है. If they talk about realistic yield then 32 होगा, ठीक है. Ideal yield is 36, realistic yield is 32. Okay. And that actually concludes our chapter on bioenergetics. Do you guys have any questions? Any questions, guys? Anyone doesn't have any questions? Anyone else? Yes, I will do it. I will upload their answers by tonight. Eamon, I have already sent these slides to you, but I'm just going to resend them. Okay. Also, they're present on the Google Drive folder. Okay. So. Okay, guys. So that's about it. Thank you, everyone, and hopefully see you on Thursday for the next class. Okay. Thank you very much, and take care.